Hello students, am I audible and visible? Yes, are we live? One minute, I think it's still loading. Yes, we are live, right? Yes, we are live, right? Okay, hi students, welcome to Biotonic Vedanto. I am Miss Gopika, your biology master teacher. So we have been doing molecular basis of inheritance for some time now. So we are going to complete the chapter today. Okay, today we have only very small portion. We have translation, HGP model, DNA fingerprinting, and lac operon regulation of your genes. Okay, so this is what we are going to understand today. So I hope all of you smash the like button and mark your attendance as well. Okay, do that quickly and let's continue. Okay, am I visible and everything? Yes, right? Yes, I'm visible. Okay, so students, the last class, not even yesterday, the last class I taught you about tRNA, right? So in tRNA, we understood that this is going to help us for translation because we learned that genetic code, right? Genetic code is there. Okay, this genetic code has to be read. There's no one else to read this code. Right now, AUG is written. Who is going to read the code? For that, we had taken the help of our tRNA, which is an adapter molecule. What is it? An adapter molecule. Okay, so adapter RNA plus the help of ribosomes. We are taking the help of ribosomes as well to read the mRNA, right? So this is the mRNA strand. These are my genetic codes. We learned this. Now on the tRNA, we understood that there is a presence of anticodon loop. What do tRNA have? It has anticodon loop. Okay, anticodon loop. This anticodon loop will form what? On the mRNA, there are some codes, right? So against that or opposite to that, complementary to that, they will form another set of it. So here, if you see, they have used GAG, right? GAG. So G will code with C, A will code with U because we are talking about mRNA, right? And G will code with C again. So CUC. So this will be formed with the help of our tRNA's anticodon loop. Now, only reading mRNA, will it help? No. Our ultimate goal, right? Our ultimate goal is to reach the position where we will be forming our own proteins, right? Where we will be forming our own proteins. For this to happen, there should be a site which will add amino acids, right? A site which will add amino acids. For that, our tRNA has an amino acid acceptor site. All of you know that? Amino acid acceptor site. Okay, this way we will be able, one side they will be reading. Okay, I told you it is like a lens, no? So one side they'll be reading, one side they will be attaching. So this is what is going to happen continuously. Okay, fine. All of you smash the like button. I cannot see the attendance. How many students are there? That many likes I cannot see. Come on, yeah, don't be conjuice. Itna to you should be able to do to your teachers. Come on, all of you. Let the attendance increase. Yes, one person listen to me. Other people also quickly smash the like button and let me know that ma'am, yes, I was present. Okay, due to some reasons, live chat is off and it is also, it will help you from no distractions. Okay, so this is clear. Now we'll try to understand what is translation. So students, translation basically according to our central dogma, DNA to mRNA we did, and it was simple because we learned transcription the best way possible. Now from mRNA we have to make proteins. Now this process is called translation. Now this process of converting mRNA to protein will only happen with the help of our tRNA, who will read the genetic code. Okay, and with the help of ribosome, they both will work together, right? Ribosome, we will be able to do this job. Okay, this will entirely become a translational entire unit. Okay, other than that, what do we need? Other than that, what do we need? We will need some enzymes, we will need some sites and all that. Okay, we will be learning that. Okay, we will be learning that. I still cannot see the attendance. Okay, I still cannot see the attendance. I'm very disappointed. I don't know why it's so much conjuicy in putting attendance. Since if you go to college, teachers will ask you for attendance. No, you will tell, no ma'am, I don't want to learn this lesson, so I won't put attendance. Right? So students, this is not a cap or a hat. This is basically a ribosome unit. Okay, this top portion and the lower portion. Now the question comes, in prokaryotes the ribosome is 70s and in eukaryotes it's 80s, but this 80s, it's basically the sedimentation unit or the Swedberg's unit. It is not depending on the size. Okay, it is not depending on the site. Is this clear? Yes. Okay, so larger unit, this is the larger unit. This larger unit is around 60s. Okay, Swedberg's unit, and the lower one is 40s. You'll be like 60 plus 40 is 100. Exactly. This is not a calculation. This is not an addition. It is basically Swedberg's unit. Now students, on this ribosome there will be 3 sites. Okay, on this ribosome there will be 3 sites. This also I told you in last class. This is like a revision. So 3 sites will be there. Shall I label it as well? So it is EPA site. EPA site. Okay, I think I should erase that previous one. 1 second. Yeah. Oh no. 1 second, students. We'll just draw the ribosome. Okay, what is it? What did I say? EPA site. Now, what is EPA site? It is very simple. A site is the site through which the amino acids, the newly waiting amino acids are waiting to come. Okay, you don't have to know in depth. You just understand so much. Okay, new amino acids are waiting. Okay, now let's write valine. New amino acids are waiting to come to the A site, and the binding site, actually the formation, the bond formation of your polypeptide will happen in the P site. Okay, so from here they will come to the P site, and E site is the exit site. Okay, so after the job is done, the tRNA will exit, right? So after this, what is going to happen, students? More people will come, and finally here if you see on the P site, 1 second. Okay, on the P side, yes, why is that in so low? Okay, on the P side there will be this formation of a entire polypeptide chain. Okay, entire polypeptide chain. Students, if you want these notes, do join on my Vedantu NEET English. I will share the notes there. Okay, Vedantu NEET English Telegram as well as on Biology by Gopika. Both the telegrams are there. So you can join for all these notes. So students, look here. This is a polypeptide chain formation. Okay. Now this is the basic of it. Now between this ribosome, right? Not only ribosome will be there. 1 second. Yes. Between this, who will be there? mRNA will be there. Right, mRNA will be there. On this mRNA, some codes will be there, and your tRNA will be there. This is the basic of translation. Is this clear? Okay, so your mRNA codon will be read with the help of tRNA plus ATS ribosomes to form the protein. That is the ultimate. Okay, that is the ultimate. So mRNA is there. Okay, on this mRNA, what is there? Codes are there or codons are there, and this will be read with the help of tRNA plus ribosome. And in eukaryotes, it is ATS ribosome, not depending on the size. Okay, finally, it will read and form the protein. This is simple as that. Now, the process comes. Okay, so look here, all of you, your ncert is here. So, translation refers to the process of polymerization of amino acid. That means many amino acids fuse together to form a polypeptide chain. Okay, got it? So, amino acids are there. It will undergo polymerization for what? For forming a polypeptide chain. A polypeptide chain. Got it? That's all. That's all. Okay? Yes. Now, the question comes. The order and sequence of amino acid are defined by the sequence of bases in the mRNA. Who will decide if valine has to come first? If, methionine has to come first? Who will decide this? That will be decided basically totally depending on the mRNA strands. Got it? Totally depending on the mRNA strands. Now, the amino acids are joined by the bond which is called the peptide bond. So, one amino acid plus another amino acid. This plus sign indicates what? Plus sign indicates the peptide bond. Simple. Plus sign indicates the peptide bond. Okay. Now, formation of a peptide bond requires energy. So, this is an energy utilizing process. That means ATP will be needed. Therefore, in the first phase itself, Amino acids are activated in the presence of ATP linked to their cognate tRNA. Do not get it confused. I'll explain it as simple as possible. Okay, yes, okay. So students, what is basically happening? Amino acids will come, right? Amino acids will come with the help of ATP. With the help of I think I'll write it in different colors. Yes. Amino acids, okay, with the help of ATP, okay, will form. First, this ATP will get reduced. Okay, wait one second. I'll write step by step. I'll write first starting to starting. But if I write here, you won't. Will it be seen? Yeah, if I write here, okay, on the top. Now, students, what is happening? First, your ATP, okay, will get broken down to AMP and 2PI. It is understood, no? It is adenosine monophosphate. Mono means single, right? Mono means single. If it has to be single, your ATP triphosphate has to release 2PI. Then only it will become single. So here you got 2PI. Okay, now this AMP is needed. We do not need this ATP. We need this AMP. Okay, so this amino acid will fuse with this AMP. Okay, can you see? Yes, you can see, right? You can see. To form an AA-AMP complex. Okay, this is the complex form. So simple, students, ATP was there. So this is called as charging of tRNA or amino acylation of tRNA. This is the process we are doing. What is happening? Your amino acids with the help of amino acyl tRNA synthetase. Okay, I'll write the enzyme name here. Which color do I write? Yes. With the help of amino acyl tRNA synthesis. Why is this needed? To break down your ATP and also to make your amino acid bind with the AMP. So ATP we broke down to AMP and 2PI. That amino acid will fuse with AMP to form AA-AMP complex. Why is this complex formed? This complex is formed because it has to bind to the tRNA. Okay, charging of tRNA will happen. Now, simple, students, this one second. I think only till there you will be able to see. Now I have to continue down. Okay, this AA plus AMP complex, right? Okay, AA-AMP complex with the help of amino acyl tRNA synthesis. This entire enzyme, okay, same thing, synthesis will get split into what? It will form AA-AMP complex. Okay, that means amino acids and AMP will suddenly, so this complex here which I formed here, no, same thing I've written here. I've just written the reaction here properly with the help of amino acyl tRNA synthetase. After this, students, this AMP, no, what will happen? This AMP will start feeling lonely. It will be like, oh, I shouldn't have come into this tRNA. So where is it going to go? It is going to go inside the amino acid receptor site. See, if I draw it like this. Okay, if I draw it like this, I cannot draw the whole thing, but I will just draw the tRNA. It will go here. Okay, after going here, it will feel very sad. It will be like, no, I shouldn't have come here. I don't want to be here and all that. Okay, so this AA-AMP plus tRNA. Now it got inside tRNA. Okay, don't forget. tRNA will form AA-tRNA complex and AMP will be out. Understood? Understood? So, if you understood, smash the like button. So, let me tell it once again to all of you. Look here, students. Let's start from the starting. So, amino acids will undergo polymerization to form a polypeptide chain. Simple thing. For this to happen, that means to form the entire polypeptide chain, they will be utilizing something called as the peptide bond. Okay, peptide bond will be happening between the amino acids. Clear? Now, the first step is charging of tRNA or amino acylation of tRNA. Okay, that means addition of amino acids. But for this to happen, we need ATP. So the first process that happens is breaking down this ATP to AMP and PI. because AMP is the one that will form the complex, ATP will not form the complex. AMP and PI. Okay, now after that, this amino acid will go and bind with the AMP and form an AAAMP complex. Simple. Amino acid and adenosine monophosphate will form a complex. Okay, with the help of which enzyme? Amino acyl tRNA synthetase. Okay, remember, amino acyl tRNA synthetase. This is very important for NEET point of view. Now, after forming the complex, this will enter the amino acid acceptor site on the tRNA. What are they trying to do? They are doing amino acylation of the tRNA complex to enter the tRNA. Then only amino acids will be accepted. Then only polypeptide chain can be formed. Okay. So, now what is going to happen, students? After going into the tRNA, this AMP will start feeling lonely and this amino acid will try to form bond with tRNA and throw AMP outside. Okay. So, once AAMP complex get into the tRNA, they will form an AA-tRNA complex and throw the AMP plus energy out. Okay, clear? Clear? Simple. Simple. It's just a basic chemistry reaction. Okay, so here, if two charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of catalyst. Which catalyst are they talking about? Amino acyl tRNA synthetase. See, I'm repeating again and again, so it is, embedded in your brains. Okay. The rate of peptide bond. The cellular factory responsible for synthesizing of protein is the ribosome. So the question can come, who is the cellular factory in translation? What will your answer be? Your answer should be ribosome. Okay. And which is the ribosome we are using? Ribosome we are using is 80S in eukaryotes, okay, and 70S in prokaryotes. Okay, in prokaryotes. Now, the ribosome consisting of structural RNAs are about 80 different proteins and it is in the inactive state. It exists as 2 units. So, students, it is in the inactive state. When will it get activated? See, once the smaller unit comes and binds with the smaller unit will come and bind with the mRNA. The larger unit will only come if tRNA is present. Otherwise, it will not come. Okay. So, yes. So, once the tRNA and the larger unit will come and bind together, it will get activated. And look here, they're talking that ribosome has a larger subunit which is around 60S and a smaller subunit which is around 40S. So, when the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. Okay. Begins. There are 2 sites in the large subunit for subsequent amino acid to bind to that, thus be close enough to each other for the formation of a, I think this got went. Okay, so the formation, wait 1 second, I have the PDF with me. Yes, so this will help in the formation of a translation complex. Clear or not? Okay. So, students, it's very simple. Do not break your head. Yeah, maybe compared to transcription, translation might look little difficult, but it is only amino acylation. Once you learn amino acylation, then it is sorted. Okay, it is sorted. Okay, so helps in the formation of a peptide bond. Formation of a peptide bond. Now, the ribosome also acts as a catalyst, that is 23S rRNA in bacteria is an enzyme ribosome for the formation of peptide. Okay, which one? The 23S ribosome in bacteria, not in humans. In bacteria acts like an enzyme. PYQ question. Okay, PYQ question. Is this clear? Simple, easy. Okay, that's all. Translation done. Okay, so here if you see this image, this is what is happening. Amino acid attachment site. For that we need who complex? We need AA tRNA complex with the help of some AMP and some energy. Okay, for this only we did all of this, and only once the ribosome comes and binds, then only the process will start. Okay, simple. Now, a translation unit in mRNA is a sequence of RNA that is flanked by a start codon, okay, and a stop codon, because mRNA does not know how long this has to happen. How will mRNA know? How will tRNA know? How will ribosome know? All of them will know only with the help of a stop codon. Okay, and mRNA also has some additional sequence that are not translated and are referred to as untranslated regions. So there are some portions which are not translated. That means which will not form any amino acids, not translated. Okay, these are called UTRs. The UTRs are present at both the ends, in the before the start codon and after the stop codon. They are required for efficient translation process. Imagine by mistake tRNA ribosome unit did not stop in the stop codon, there will be UTRs which will actually not help in translation, because of which automatically the system will switch off. Okay, now for initiation, the ribosome binds to the mRNA at the start codon that is recognized only by the initiator tRNA. Students, very important for your translation. Okay, for your translation to happen, we need someone called as initiator. Okay, initiator tRNA. This initiator tRNA will only help in the initiation. That means will actually help in binding with the start codon. Will help in the binding with the start codon. So initiator tRNA is actually going to help. Now the ribosome proceeds to the elongation phase of the protein synthesis during the, so here also if you notice, there is actually an initiation, there is an elongation, there is a termination. But the thing is, it is not very clear, like it is not written so, you know, what to tell, it is not mentioned so much that we are not writing about it. But understand, initiation will be done by initiator tRNA. Okay, elongation step, what is going to happen, students? Here, so in the elongation step, what is going to happen? Here the formation of, okay, formation of polypeptide chain. Simple. Formation of polypeptide chain will happen. Correct? Okay. Then in the termination will happen. Okay. The ribosome proceeds, and during the stage, complex composed of an amino acid linked to the tRNA sequentially bind to the appropriate codon. See, what is it? Amino acid linked to tRNA. This is why we did amino acylation. Is this making sense now? Okay. Now, the ribosome moves from codon along the mRNA. So, how it is, you know, now I have a rows here. Let's say. Okay, students, this is the tRNA. Okay, can you see? 1 second. Okay, this is the tRNA. This flower is an mRNA. Okay, this tRNA and ribosome, everything. So, how are they going to do? They are going to read like this, right? They are going to read like this, read like this, read like this, read like this. By the time here, polypeptide chain will be continuing to form, longer and longer and longer it will get. Okay, that is what is happening. Clear? Yes. Okay, fine. Yeah, now 1 second, students, I need some water. One second. Okay, so sequentially bind to the appropriate codon in the mRNA by forming complementary base pairs with the tRNA anticodon. Now the ribosome moves from the codon along the mRNA, and amino acids are added one by one. It is not like everything is going to come and form like that. So what is going to happen? Yeah, what will happen? From the A side, it will go to the P side. In the P side, formation will happen. From there, the tRNA will exit from the E side. Okay. Now, at the end, okay, at the end, that is during the termination, there is a release factor that binds to the stop codon, terminating translation. So in translation, let's revise once again quickly. Okay, let's revise once again quickly. So in translation, okay, first thing was initiation. First thing is initiation. In initiation, what happened? First, before initiation only, your amino acylation happened. Okay, amino acylation of tRNA. Okay, thank you so much. Yes. Okay, so amino acylation of tRNA happened. Now during this amino acylation, what did we form? Amino acid plus tRNA complex we formed. Okay, after that we have to wait for tRNA, initiate the tRNA. Okay, we have to wait for, please write it with me, do not wait for the notes, write it with me. Initiate. Students, if you learn this chapter, you are going to get 40 marks in your NEET. Okay, 36 to 40 marks guaranteed from this chapter. Okay, so learn it with me. Initiate the tRNA, which will start initiation. Okay, after that, when we go further, next one will be elongation. Okay, next one will be elongation. In the elongation, what is going to happen? Here we are going to form the polypeptide chain. Okay, polypeptide chain. So why is this, how is this polypeptide chain formed? Polypeptide chain will be formed by forming this complementary, okay, complementary codons, and that codons will tell us which amino acids to bind with. So amino acid binding, and then formation of peptide bonds. Okay, then during the last stage, that is your termination, what do we have? We have the presence of stop codon. Okay, other than the stop codon, we have release factor. Okay, other than the stop codon, we have release factor that will actually terminate the reaction. And at the end, before the start codon, that is before AUG, there are some UTRs, and after the stop codon, say UAG, there is some UTRs. This way, the entire translation will come to an end. Is this clear? If it's clear, come on, smash the like button, all of you. Yes, clear? Easy? Yes, easy? Okay, fine. Done? Next topic, shall we go? The translation done. So students, here if you see, here this is your NCERT diagram. Okay, here you can see what is happening. See, one person is waiting, no? Where are they waiting to join? They're waiting to join from here. tRNA is waiting. Okay, tRNA is waiting. This is going to come to the A side. Okay, this where the bonding is happening will be the P side, and then where the rest of the polynucleotide, growing polynucleotide will be going, right? This will be the E side. After, no, here this is the E side, this is the P side. Okay, this is the P side. So here they have shown two of them. Can you see? One is going to, this is going to come and join. So this is waiting to come. This has finished the process and it's exiting, and this is the growing polynucleotide. Here if you see, this is the smaller unit of ribosome, this is the larger subunit of ribosome. Only once this both come and fuse, the translation will ever happen. Easy, right? Easy, right? Now students, regulation of gene expression. It is a very simple concept. Let's try to understand very simple. Students, regulation means I am able to control something. Now switch off or switch on of something. That means what? Now I have switched off the live chats. How did I do that? It is because I'm able to do some kind of controlling. Yes or no? So for that to happen, I think I had 1 second, students. Yes, see, I had put the NCERT lines, but somehow it didn't show. Okay, so regulation of gene expression refers to a very broad term that may be similar at various levels. That means an organism or a gene, if it feels like, okay, I don't want to do transcription, like I don't feel like doing transcription, I want to switch off. That means students understand, every time you don't have to be active, right? Every time you don't have to be continuously talking. Sometimes you will be like, I want to sit peacefully, I want to sleep, I want to watch some series. So there are different types and modes in which you want to work. Same way, even the genes, sometimes they don't want to be switched on, they want to get switched off. Now, every time they cannot get switch off, not at every level they can get switch off, right? Can you sleep every time of the day? No, you can't, right? If you're working, there are some time when you get a rest, only that time you can take a nap or anything, right? So same way here also, students, during the transcriptional level, okay, during the transcriptional level, the gene can decide to get switched off. That means formation of a primary transcript. So during transcription, they can get switched off. During the processing level, that means simple RNA processing, all of you learned it with me. What is this? Your splicing, right? During the removal of introns, during the addition of capping and tailing, during that stage also, if the gene is feeling like, no, I don't feel like I should process, I will switch off. Okay, this is regulation of the gene expression. Gene is controlling some things, okay? And then transport of mRNA from nucleus to the cytoplasm. During the translation initial stage also, they can switch off. Okay, translation initial stage where mRNA from nucleus has to go to the cytoplasm so we can form proteins. Yes, during this stage also, the gene can decide to stop. So during the translational level, during the transcriptional level, during the RNA processing level, all these places, the gene can decide to switch on or switch off. Clear? Okay, gene can decide to switch on or switch off. Okay, so the first site of regulation, students, where this question was asked, I think in NEET 2020, first site of regulation in genes. First site of regulation in genes. Where is that happening? In the transcriptional level. Okay, that should be answer. Is that clear? Yes. So first site of regulation. First site of regulation is in the transcriptional level. Okay. Done? Okay. Now, we spoke about gene expression, but thank God you people do not have gene regulation in eukaryotes, which is a blessing, because eukaryote gene regulation is very, very complex, complex process. So here you only have to understand for prokaryotes, and out of that also you do not have to learn all the regulation. You only have to learn lac operon concept. Okay, so regulation of gene expression, first site of regulation, a PYQ question. Please make a note. Now, lac operon. Students, lac operon is a very simple thing. It is basically in the presence and absence of lactose. Okay, lactose is present or lactose is absent. So depending on this, the gene will get regulated. Okay, depending on this, the gene will get regulated. So it is simple, students. There is a regulatory gene. Okay, there is a regulatory gene. Okay, regulatory gene and some. So what is happening is one person will control, the rest of the people will do the job. So same way, your regulatory gene or your I gene will release something, okay, and that one, okay, so what is it going to do? It codes for repressor protein. What is repressor protein? What is repressor protein? Repressor protein is something that will form repressor. That means this repressor will go and bind with the operator and stop or switch off the gene. Now, simple, do not worry. Be like, ma'am, what if I do not know anything? Okay. Pipo. Can you remember this? Pipo ZYA. What is it? Pipo ZYA. This is the gene regulation. Okay, so we have a promoter region, we have an I region, inhibitor region, again we have a P region, O region is the operator region, and the ZYA region, right? The ZYA region will release some set of enzymes. These are LAC-Y, LAC-Z, LAC-Y and LAC-A. These will release some kind of enzymes which will actually help in lac operon concept. Okay, which will help in lac operon concept. Is this clear? Visible? Okay. Okay, fine. Okay, so what are they? They are LAC-Y, LAC-Z, LAC-Y, LAC-A, and Pipo. In this I gene, this I gene will code for repressor protein and release repressor. This repressor will go and bind with the operator. Now imagine RNA polymerase is happily coming to do transcription. Okay, RNA polymerase is coming like this, saying that, yeah, I'm going to do transcription, I'm going to form mRNA. It is coming like this all the way, but as soon as it reach near the operator, it will realize that there's a full block here. There's a block. Who has block? Repressor has block. So RNA polymerase will get disappointed and RNA polymerase will stop the transcription. Now this way, this is the first site of regulation I told. This way the gene has decided to switch off. The gene has decided to switch off. So what happened? RNA polymerase was coming. Why will RNA polymerase come? To do transcription, right? It is the first site of regulation. So it was coming to do transcription. So RNA polymerase kept coming. As soon as it reached the operator, it realized that this operator is got blocked. Okay, I'm not able to see the operator because the repressor came and bound with the operator. Now RNA polymerase cannot move further. By this method, gene got switched off. Okay, now how do I switch on the gene? So this whole thing is happening when the lactose is absent. Okay, so here we are talking about prokaryotes. So here we are focusing on E. coli bacteria. Imagine E. coli bacteria had enough of glucose in its body. Then lactose will be less. So during this time, switch off. Gene will be switched off. Okay, and imagine the E. coli did not get any amount of glucose from anywhere, and because of that lactose it has got. Lactose is more, glucose is less. Gene will switch on. Gene will switch on. Now how? Simple, students. Again, Pipo, let's write. Pipo ZYA. Okay, who is coming happily for transcription? RNA polymerase is coming. RNA polymerase is coming. Here again it will code for repressor protein. Okay, this fellow will again code for repressor protein and it will form repressor. But in this case, lactose is present. Lactose, or your inducer according to your NCRT, will come and bind with the repressor protein, not allowing the repressor protein to go and bind with the operator. Now RNA polymerase will come happily, do the transcription, all the way it will move from the operator, and these three people will release one set of enzymes which will actually help in switching on the entire process. Okay, actually students, lac operon concept is a little leaky process. That is the reason why you will see a presence of lactose here. Okay, because lactose we saw here, no, but still here also we will see some presence of lactose because your lac operon concept is a leaky process. That means some leaks have happened. Okay, so now the gene will get switched on. Is this clear to all of you? Okay, now the gene will get switched on. Let's try to understand what did I say. Okay. Okay, now let's talk about lac Z, lac Y, lac A. These three are there. Lac Z will release something called as beta galactosidase. Release called something as beta galactosidase. Okay, and lac Y and lac A. Lac Y will release something called as permease. Okay, and lac A will release something called as trans-acetylase. Trans-acetylase. Okay, now beta galactosidase is used to break the lactose into glucose and galactose. Clear? Okay, this is why I told you it is a leaky model. Why did I tell it is a leaky model? Because here lactose is there. Lactose was there. That is why RNA polymerase reached up to lac Z, no. But then how come here lactose is there? So that is why I told you it is a little leaky model. Okay. Now permease, students, permease will actually help in increasing the permeability. Increasing the permeability of your E. coli. Okay, E. coli cell. That means imagine this is your E. coli. Oh, E. coli became like, oh no. Okay, it's okay, happens. Okay, so once again let me write lac Z, lac Y and lac A. Okay, lac Z will release beta galactosidase, okay, which will break down lactose to galactose and glucose. Okay, and lac Z will release permease, which will increase the permeability. of the cell wall. Okay, of the cell wall of E. coli. Now what I was trying to show, imagine this is an E. coli. Okay, this is an E. coli. This E. coli needs to absorb more lactose. If it only absorbs more lactose, will the gene get switched on? Will E. coli be able to do transcription? For that to happen, this permease will increase the ability of this cell, right, to take in more lactose. Okay, if it takes in more lactose, then easily lactose will be broken down to glucose and galactose, and this E. coli will have glucose enough for energy. Okay, and the last one is LAC-A, which will release trans-acetylase, which function is still unknown. Okay, function is still unknown. It might help in one of these processes, but the function is still unknown. So only if the operator gene is free, right? Only if the operator is not bound with the repressor, then these people who YA and these people will get active. Okay, they'll get active. If operator is bound with, it's not bound, it is bound with repressor, then these people will be not doing any job, will be inactive. So during this first stage here, who should be there? This repressor will bind with lactose, that is your inducer, and here repressor will not have no lactose. Okay, no lactose, glucose will be there. This will switch on the gene, this will switch off the gene, and switching off will happen in which stage? In the transcriptional situation. Okay, will happen in the transcription. Easy? Easy? Yes or no? Easy? Okay, let's see your NCERT diagram. You will understand thoroughly this. Okay, students, look here. Here, Pipo ZYA. So here you can see I gene is forming the repressor mRNA. That repressor mRNA will form repressor. This repressor will go and bind with the operator region and prevent the RNA polymerase from doing transcription. Okay, this is in the absence of inducer, or basically in the absence of lactose. Okay, now here you have Pipo ZYA. Again, repressor mRNA will form repressor. Same thing will happen. But here what is happening? Lactose is present. Lactose is present. So this inducer will go and bind with the repressor, making the repressor inactive. Now what is going to happen? This repressor mRNA, RNA polymerase will happily come and form mRNA. Transcription will happen, and the release Z will release beta galactosidase, permease, transcriptase, and after translation, transcription also will happen. Easy, lac-operon? Lac-operon is easy, no? Okay. Now HGB project. So students, if I have to talk about HGB project, I have to give you a little idea about HGB, right? So HGB means, the full form, Human Genome Project. Human Genome Project. Okay, this Human Genome Project, if I have to talk about, was a very, very, very mega project. Mega awards and all you have seen, no? That means high budget, right? Mega project. Why was this done? Okay, and by whom was this done? Okay, it was done by James Watson. It was done by James Watson. Okay, clear? James Watson. And when did this take? This took almost from 1st October 1990 to 2003. This took almost 13 years to come in existence properly. Okay, it took almost 13 years for this project to be a successful. And not only this project is not called mega project just because of the budget, but it is also called mega project because of how many other big institutes joined hands with this project. Okay, like a department of in Japan, all the top departments of Japan, France, Germany, US, all these people came together to make this project a successful one. Okay, and this, I told you, mega project, no? It was the budget of this project. Okay, the budget of this project was $9 million. Okay, $9 million was the budget of this entire project. Okay, what was this project aiming for? It was aiming to identify all the 10 into 9 base pairs of our human genome. Okay, and it was also helping in to avoid the storage books, right? Imagine you have to get this 3300 or your 3 into 10 to the power 9 base pairs in a book. Imagine how much it will cost us, right? It would be 1,000 pages book. Like that, we would have had 3,300 books. So to avoid that, to have no books for our genome, to understand where mutation happens, okay, to understand or learn or understand the genome of a human, for all this HGB was useful. Okay, so some important goals of HGB were: identify all approximately 20 to 25,000 genes in a human DNA, determine the sequence of 3 billion base pairs that make up the human DNA, store this information in databases other than in books. Okay, not in books. If you store it in books, it would have taken some 3,300 books per person. Okay, improve the tools for data analysis. This way we will be able to understand our diseases better, mutations better. So many things would be understood better if we have an entire database of our genome. Okay, transfer related technologies to other sectors such as industries to develop better equipments, medicines, drugs, and etc. Now, address the ethical, legal, social issues. Students, this one's full form can be asked as a PYQ. Okay, the sorry, MCQ. Okay, this full form can be asked as a MCQ. Clear? MCQ. Address the ethical, legal, and social issues that may arise from this project. Okay, to understand all of this, then salient features. So this HGP goals and salient features you have to by heart by yourself. There is no other method. Okay, there is no other method. You have to learn it by yourself. Okay, students, the human genome contains 31647 billion base pair. How much? 3164.7 billion base pair. The average gene consists of 3000 bases, but the size vary with largest known gene called the dystrophin. Very important. Okay, dystrophin, which is around at 2.4 million bases. Okay, the average gene consists of 3000, but then it varies. That is, the largest human genome is dystrophin. Okay, dystrophin. The total number of genes is estimated at 30,000, much lower than the previous estimate. So before, without even knowing this HGB, people used to be like, oh, maybe we have around 80,000 to 1,40,000 genes. But then after they understood this HGB, they realized that the total number of genes is estimated at 30,000. Okay, 30,000. Now, the functions are unknown over 50% of the discovered genes. Only 50% genes, we have an idea what these genes are doing actually. Okay, less than 2% of the genome codes for protein. So out of these, only 2%, out of the 50%, only 2% will code for proteins or do translation. Okay, or do translation. Repeated sequence make up a very large portion of the human genome. That's why, students, we all have around 99.9% same DNA. That is called the bulk DNA. And what is different? 0. Sure. Okay, we have hardly 1%, 0.1% is only makes us different from each other. Okay, only this much. Hardly not even 1% makes us different from each other. Okay, now salient features of HGB we understood. Now students, look here. A person cells, chromosomes, that will be, the entire base pairs will be into a database. Now students, two methods were there to do HGB project. Okay, what are the two methods were there to do HGB process? First one was like a sequence annotation. and second one was expressed sequence tags. Okay, what is it? Expressed sequence tags. Sequence annotation and express sequence tag. This was two methods by which they could do HEB further. One was a blind approach, one was a better approach. So I'll tell you what it is actually. Okay, it is very simple, students. So in the sequence annotation tag, they used a method of taking the entire DNA sequence with both coding and non-coding. They took the entire approach with coding and non-coding, and they also took one more approach, which was amino acid sequence. This was a blind approach. Why did they call this a blind approach? Because they took both coding and non-coding. It did not make sense, right? They took everything, coding, non-coding. So what is the use of non-coding, right? Taking non-coding. So it was a blind approach. Next is expressed sequence tags. Express sequence tags. Sorry, students, 1 second. Okay. So yes, express sequence tag, students. What did we do? Here we extracted the mRNA. Okay, extracted the mRNA. By this method, what we were ultimately doing is we were taking only the coding strands. And not the unnecessary part of the DNA. Okay, clear or not? Okay, smash the like button if you understood, all of you. Okay, this way we extracted the mRNA, and with that extraction we were able to get only the coding strand, and this was a better approach compared to your sequence annotation. Okay, it was a better approach compared to a sequence annotation. Now what are the host and vectors they used? Host was simple, bacteria and yeast. Okay, and the vectors was BAC and YAC. That is bacterial artificial chromosome and yeast artificial chromosome. Okay, clear? Okay. Now students, the last topic of this chapter, DNA fingerprinting. But before that, I think I have to tell you about SNPs also. This I'll tell it here. Okay. Students, SNPs is there, no? That is mentioned in your salient features. If you see, SNPs are mentioned. SNPs are nothing but, students, single nucleotide polymorphism. Single nucleotide polymorphism. What is single nucleotide polymorphism? Now imagine I have straight hair, even Banod or Rani has same straight hair. Now the difference between Rani and me is one single nucleotide would have changed. Okay, one single nucleotide would have changed. Now say, let's say Rani, okay, Gopika. Now let's write some random series. 1 second. Okay, G, A, C, A, G, C, G. Okay, here also G, G, C, A, G, C, G. Now what is happening, students? Here can you see? This one nucleotide difference will make both of us completely different. Texture of the hair, looks, eyes, everything. One small thing can make a complete difference. Is this clear? Okay, is this clear? So that is called as single nucleotide polymorphism, because of which, again, you know, sickle cell anemia, point mutation. So all this can happen because of that. Okay, all this can happen because of that. Okay, sickle cell anemia is because of one small change in the nucleotide. Okay, HbA gene will be replaced by HbS gene, because of which sickle cell anemia will be caused. Okay, yes, done. Done. Okay, fine. Let's go. Now, DNA fingerprinting, last topic of this chapter. DNA fingerprinting, students, it's basically helping us. Okay, it is basically helping us with with lot of crimes, especially with lot of crimes, and also with a lot of identification. If a child is missing, with DNA help we do identification. All this is helping. Now, how is DNA fingerprinting helping? It is simple. Students, understand, I told you bulk DNA or junk DNA. The DNA which is common for all of us will be called as junk DNA or bulk DNA. Bulk DNA. Now, this bulk DNA, students, will be completely repetitive strands. Yes or no? Will be repetitive. Oh, repetitive become too many repetitive. Repetitive. Okay, repetitive sequence. Okay. Now, students, this repetitive sequence, okay, most of this repetitive sequence will be non-coding sequence. Okay, non-coding sequence. So, what is this DNA fingerprinting helping is? Basically, it will help us in DNA profiling. Okay, DNA profiling. Now, look here, let's say repetitive DNA, here they have told, see, involves identifying differences in some specific region in the DNA sequence called as the repetitive DNA. So, I told you this repetitive DNA will be so many of them, okay? Like this, it will keep continuing. But in this, we have to understand which is the sequence which is different, okay? Because in this sequence, a small stretch of DNA is repeated many times. So, imagine now this AAG is repeated for me around 10,000 times, and for Rani, it will be repeated 1,000 times. So, this will make both of us very, very different from each other. It is not necessary that for everyone, the same repeated sequence will be repeated the same number of times. If it is repeated 10,000 times for me, for another person, it might be repeated only 10 times, 1,000 times, 100 times, okay? So, that is what we have to identify with the help of DNA fingerprinting. These repetitive DNA are separated from the bulk genomic DNA as a different peak during the density gradient centrifugation. So, density gradient centrifugation will help us understand different peaks. Okay. Yes, clear. Fine. The bulk DNA forms a major peak. Obviously, students, bulk DNA is 99%. So, this will form a major peak. Okay, 99% will be bulk DNA. And the small peak are referred to as satellite DNA. So, the DNA which is helping us in making each other different, that is called as satellite DNA. Okay. Depending on the base composition, AT and GC rich, length of the segment. So, what and all are we looking into? We are looking into the base composition. That is, there are regions which are AT rich, there are regions which are GC rich. Usually, they say that GC rich regions are around 60% in our DNA. Okay. And AT rich regions are only 40%. So, depending on this, your micro, mini, satellite areas. Okay, satellite DNA is again divided into micro satellite and mini satellite. It's a part of the chromosome. Okay. Now, length of the segment, number of repetitive units. The satellite DNA is classified into many categories such as micro satellite and mini satellites, etc. I told you, this is what I meant. Now, these sequences normally do not code for any protein. So, what are they trying to say? They are trying to say that this repetitive DNA is usually non-coding DNA. Non-coding DNA. Okay, important. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Okay, since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism. That's the only reason, even after a rape case, even after a murder case, even after a robbery, even if they find a hair, follicles, nail, sperm, anything, they will be able to still find the DNA. That is the reason why people do nowadays a lot of, unnecessary things. They do like removal of DNA fingerprinting. Like if they have touched some object, they'll use a sellotape and remove that, which is shown in movies nowadays very commonly. That is the reason why it is difficult to find the culprits nowadays, because of how much technology is there, right? So everything will have the same degree of polymorphism. So we do not have to worry that, oh my god, I only got a hair from a crime scene, I should have got saliva. Nothing like that. Everything will have the same degree of polymorphism. They become very useful identification tool in forensic application. Further, as polymorphism are inheritable from parent to children, DNA fingerprinting is the basis of paternity test in cases of abuse. This happen a lot in abroad, places like US, UK and all. A lot of this happens, telling that this is my child. They kidnap the child and then they will, tell that it is my child, right? That things happen a lot. Okay, now students, how does this DNA fingerprinting happen? It basically happens in 6 steps. Okay, basically happens in 6 steps. First one is isolation of DNA, because I told you we have a lot of junk DNA and we also have a lot of necessary DNA, that is junk DNA plus your satellite DNA. We need to remove out what we need, right? We need to remove out what we need. Digesting the DNA by restriction endonuclease. What is this restriction endonuclease? Biological scissors. Biological scissors. Okay. Now, separation of DNA fragments by electrophoresis. This is to help in band formation. What formation? Band formation. This will help us understand which are the ones which are, where is each of them present, and this will help us identify the rest of it. Okay, now students, separation of DNA fragments by electrophoresis, is this clear? Yes, it is seen. Okay, now before this step, okay, before restriction, after this restriction endonuclease, sometimes we use a density gradient. Okay, I'll show you that. One second. We use a density gradient, that is your with the help of CSCL, which you studied in your Michelson and Stahl experiment. You will get 2 bands. You will get 2 bands. This will be your main DNA. Okay, this will be your main DNA. And this will be what? Repetitive DNA. Will be your repetitive DNA. Okay, this will be your main DNA, which is non-repetitive and which will be coding. Which will be coding. This will be your repetitive non-coding sequences. Okay, this way what will happen, students? We'll be able to get a different gradient one. Now this repetitive DNA usually are AT rich DNA. This is GC rich region. Okay, GC rich region. Keep it in mind. AT rich region and GC rich region. Okay, clear? Clear? I think it's visible, no? Yeah, it's visible. Yes. Visible, right? Yeah, visible. Okay, continue. Okay. Now students, look here. Digestion of DNA by restriction endonuclease. This is for chopping, whatever we need. Then separation of DNA fragments. That is basically like this, students. This is your gel electrophoresis. Okay, some numbers will be given. Say 1 to 11. Let's write as of now. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Now what will happen as we run this? Okay, as we run this, depending upon the gradient, some will settle here, some will settle here, some will settle here, some will settle here. Like this, it will keep moving. Okay, this is your gel electrophoresis. Okay, this is the formation of band. Okay, 1 second. Yes, this will be run on an agarose gel plate. Okay, agarose gel electrophoresis. Agarose gel electrophoresis. Okay, after getting this gel electrophoresis result, so what are we actually focusing? If you look at your NCRT, you will all be able to see one diagram, right? Yes or no? All of you can see a diagram. Okay, you'll be able to see a diagram where they're showing suspect A, suspect B, and the blood sample we got from the crime scene. Okay, so this will help us understand how many repetitive DNA did each of these people have, and which is the blood matching to the one that was there in the in the crime scene. Okay, after this, student, this gel electrophoresis, we are not supposed to move. So from that, we are going to transfer to a blotting paper. Okay, what are we going to transfer it? Transfer it to a blotting of separated DNA fragments to a synthetic membrane such as a nitrocellulose or a nylon membrane. Okay, to a nitrocellulose or a nylon membrane, and this is called as southern blotting. This is called as southern blotting. Okay, done. Now here, what are we after that? So here your basically double-stranded DNA will get denatured to your single-stranded DNA. Okay, DNA will get denatured. DNA will get denatured. Okay. Then what is happening? Transfer DNA from your gel electrophoresis to a blotting paper. Okay, gel electrophoresis to a blotting paper. After that, we are using something called as VNTR probe. Students, yes, we understood that depending on the density gradient, we'll be able to understand. Okay, but what is this VNTR probe? This is actually variable tandem repeats. Okay, variable tandem repeats. Now, students, what is this variable variable number of tandem repeats? N stands for number. Variable number of tandem repeats, students. These are complementary strands. These are complementary radioactive strands which are formed for understanding this. Now, imagine every person will have a set of satellite DNA which makes them different, which will be like from the maternal and paternal, right? That DNA complementary we will form so that this VNTR can hybridize using a VN. That means after getting this southern blotting value, this VNTR probe will go and bind with, imagine, VNTR probe will go and bind like this, and what will happen, because of which we will be able to understand who is the suspect. Okay, detection of hybridized DNA fragments by autoradiography. Can you understand? Autoradiography. Here this VNTR probe was radiographed, right? Radioactive. So after that we will be able to see in autoradiography a systemic representation was shown below. I think I did not put the systemic representation. 1 second, students. 1 second, I'll show you that also. Can you all see this? All of you look here. This is given in your NCERT. Yes, can you all see? 1 second. Yes, you can see, no? So students, look here. We'll try to understand what did I just say. I cannot write. That's the only problem here. I cannot write. Okay, students, look here. Paternal chromosome. Okay, paternal chromosome. Here it is 3. Okay, here it is 1, 2, 3, around 11. Okay, maternal chromosome, here it is 4. Here it is around 9. Okay, now what is happening? Then we have chromosome 2, which are almost similar. Chromosome 16 is there. Now DNA from individual A and DNA from individual B. Now this is the thing that we got from the crime scene. Okay, we got this one from the crime scene. Here if you notice, you can see that there are 11 paternal DNA, 9 maternal DNA, 7 and 2. Okay, chromosome 7, chromosome 2 and chromosome 16 is there. Now if you look here, can you see that this C gel electrophoresis, C gel electrophoresis, shall I zoom it a little more? Yes, I think it will be visible now. Yeah, C gel electrophoresis and A and B, if you see, B and C is matching completely. Okay, can you see how the band formation happened? Okay, so we will be using VNTR probe to go and bind with these people so that under autoradiography we will be able to highlight this. We'll be able to highlight this. Okay, so amplified repeats separated by the size of on the gel. So this person's will not see, 2 people are there, but this person's will not match this person, nor this person's will match this person. So here if you see in the crime scene, okay, crime scene, we got blood sample C. And that is exactly matching with blood sample B. Is it easy? Okay. Okay, so we will be using radioactive VNTR probe, which are complementary to the single-stranded DNA. They will correctly go and bind, and under the autoradiography, we will be able to see the highlighted portion. Okay, so when we do the film, we will be able to see that auto radiography, and we can see dark bands on the X-ray film. These bands which you're seeing, no, these are all under auto radiography for us to get a clarity on it, for us to understand, yes, if this is the person who has done the crime or not. Easy? Okay, easy or not? Yes. So yes, students, 1 second, let me go back to our PPT. Yes, so what and all did we do in this? We did first, we did here, where to write? Yeah, yes, we did DNA fingerprinting. First we did separation. After that we did DNA on gel. Okay, gel electrophoresis. After that we transferred that into southern blotting, that is on a nitrocellulose membrane. We did southern blotting on nitrocellulose membrane or your nylon sheet, and from that we used VNTR probes, which are complementary. What do I mean by complementary? Now imagine ACGAATT. Okay, now what will happen? This VNTR probe will form complementary. For A it will form T, for C it will form G, C, T, T, A, A. This will go and bind, and under auto radiography we will be able to see dark bands. Auto radiography we will be able to see dark bands. This way we will be able to understand exactly, because on the gel electrophoresis it will not be that visible, it will be slightly whitish color. So what will happen, we will not get a clarity on that. So for that we will use VNTR probes, which will go and bind with the single stranded DNA, and it will give us the proper dark band under the auto radiography. Okay, if you have seen this UV rays and all that, no, you would have seen, I think now you wouldn't have done it, but yeah, you will see all that later in your, okay, later in your classes. Done? Is this clear? So yes, students, that is all about your molecular basis of inheritance. I hope you understood. Little complex portion is your DNA fingerprinting and HGP, so read it multiple times, otherwise you will not understand. But other than that, lac operon, all of that concept is very easy. It is not that difficult. Even translation, only this equation is actually an extra information, because it is not mentioned in your textbook. But when teachers explain, we always try to make you understand, because amino acylation, students will not generally understand by the word. So we try to make them understand by this equation, which is a three-step equation. So yes, if you understand this equation, then you're good to go. And if you remember the enzyme name, it is great. So yes, students, that's all. And if you are students who are preparing for boards and is very, finding biology very difficult or want help with biology, do join us on Vedantu NEET English, because we have a total solid dedicated plan for boards. It is going to help a lot. So yes, do come and subscribe to our channel and join us on our Vedantu NEET English platform, because I will not be able to take two classes. So I'll be fully there. I'll be taking long marathons there. So next week we will be preparing all of you for boards very well. So all of you gets full 70 out of 70 for your CBSE and for your PUC exam. So yes, students, thank you so much for joining. And also there are a lot of links in the description. If you notice, the first link is for the crash course. If you want to join the crash course, do join the crash course. And the code to use is GGCC, where you will get the entire crash course for 4,000 rupees. Okay, the code is GGCC. The entire crash course is for 4,000 rupees, where we will complete the entire 11th and 12th syllabus. I will not be teaching that. Ritu ma'am will be taking. Ritu ma'am, if you know Ritu ma'am. Okay, she will be taking the zoology part. All experienced teachers with 8 to 12 years experience. So yes, do join our crash course. It will help you a lot. If you are a student who is needing that last minute push, then this is the batch for you. This is the crash course for you. So yes, do join the crash course. Click on the link. And we also have a NEET treasure box, where you will get all the PYQs, chapter wise PYQs, notes, links, everything on that NEET treasure box. Just click on the link in the description box and join us on the join us there, and also join our Telegram, where you'll get all the updates about the whatever NMC, NTA, whatever they do, everything we will let you know on the Telegram channel. Okay, so yes, students, thank you so much for joining. Any doubts, please feel free to put it in the comment section. And that's all. Bye bye.